The nutrient media used for the culture of animal cells and tissues must be able to support their survival as well as growth. They provide nutritional and hormonal factors. There are two types, natural and artificial. Natural media consists solely of naturally occurring biological fluids like coagular clots, that is plasma clot. Biological fluids like amniotic fluid, acetic acid, pleural fluid, serum, aqueous humor of eye, insect hemolymph, etc. Tissue extracts like chick embryo extracts, spleen, liver, bone marrow extracts, etc. They are used for organ culture. Artificial medium with or without serum are used for cell cultures. Example, serum containing media is Eagle's minimum essential medium with 5 to 20% serum. Serum provides the plasma protein Delbecos. Enriched modification of minimum essential media DME is serum-free artificial media. Initiation of cell culture. This involves 4 steps, that is preparation and sterilization of the substrate, preparation and sterilization of medium, isolation of the explant. The site of opening is sterilized with 70% alcohol and tissues removed aseptically and placed in a balanced salt solution. Then we disaggregate the explants. Cell cultures are generally started from disaggregated plants. Disaggregation is by mechanical method or enzymatic method using trypsin, collagen, or using EDTA. EDTA means ethylene diamine tetraacetic acid, which is used for disaggregation. Next is subculturing. When mechanically or enzymatically disaggregated, then tissue pieces of cell are cultured. They attach to a substrate and monolayer of cells develop from them. They are called primary cultures. After a period of time, the available substrate becomes occupied by the monolayer. Cells are then harvested from the primary culture and inoculated or seeded on to the fresh vessel. The cultures thus obtained are called cell lines. They can be propagated, characterized, and stored. The term cell line implies the presence of several cell lineages, either similar or distinct. Among these cell lineages, if a particular cell lineage has specific properties that are identified in bulk of the cells of the culture, it is then described as cell strain. A cell line or cell strain may be finite or continuous depending upon whether it has limited culture lifespans or if it is immortal in culture. Finite cells grow up to 20 to 80 population doublings before extinction. Now, every cell line is given a code, that is, for example, NHB, that is called as normal human brain, and a cell number which, when several cell lines have been divided from the cell source, it can be NHB1, NHB2. A number of population doubling. The cell lines have already undergone 2 by 2 means 2 doubling. The process of inoculating cultured cells into fresh culture vessels are called as subculturing. Subcultures may be done every 7 days, but some fast-growing cell lines require a change of medium after 3 to 4 days of feeding. The uses of using cell culture is large-scale. Animal cell cultures have been used in recent years as a source of valuable products like enzyme, hormones, interferons, monoclonal antibody, and vaccines. Before that, I like to mention about a cell line called Henrietta Lacks cell line, that is HeLa cells, which was taken from an African American woman of 31 years old who was suffering from cervical cancer. So her cells for biopsy cervical cells were taken, and it was found to be it could be kept live. It was found that it could be kept live in vitro, and but sadly she passed away without knowing it. So, but still till date we use HeLa cells as a immortal cell line.